Now that you have your samples in their individual and labeled tubes, you're ready to begin with the extraction. What we'll need to do is add 100 microliters of the nucleolysis solution. So I'm going to use my blue pipette, set to 100 microliters. And always, I want to use a fresh tip to add the solution every time. My lysis solution is already on ice, so I'll carefully get 100 microliters, cap that back up, and add that 100 microliters to my first sample. Once you've added the solution to the sample, you can take a clean pestle and start grinding the sample forcefully for about 1 minute. Once you've ground the sample thoroughly, and in some cases you may notice that the solution changes color. Chlorophyll might be released with plants. With animals, you may see some color change. Just grind it until you hopefully see some of that color comes out. That means that you've done a good job. And then add the remaining 500 microliters of the nucleolysis solution. Make sure to mix after you added that last 500 of the nucleolysis solution, and add this to the heat block. Once your samples have incubated for 15 minutes at 65 degrees, you have to add the RNase solution. The RNase solution contains RNase enzyme, and that enzyme is going to actually break down a lot of the RNA that's present in the solution, so that when we actually precipitate nucleic acids, the majority of what we're going to get back is DNA. I'm taking my P10 and I've set it to 3 microliters. And you want to take a fresh tip, take up 3 microliters of the RNase solution, and add that to each one of the tubes. Once you've added it to the tube, you want to shake the tube and then place it at 37 degrees. So if you have another heat block, place it in that one. If you need to let this heat block cool, just cool it to 37 degrees and then place this to incubate for additional 15 minutes.